So just keep in mind that you have time to do it, but it's better for you if you do them early. One of the things that I've noticed about some of the ways in which you're working with Quirkus right now, some of you are not logging on at all, period, and that's not great. So you should try to log on at least once a week just to make sure that you're up to date on some of the announcements, and that will help with some of the different types of questions that you're asking via email as well. Today's lecture is going to require that in several places that you're going to have to take down some notes, so just to keep that in mind. So are we good now? Okay, awesome. Thank you, gentlemen. Okay, so what we're going to start doing is going back, re-examining a couple of things related to the unicorns, things that we're not 100% sure of. Do they actually exist? How would we prove that they exist? What's the controversy about them? And what we're going to be using as a framework is everything that we've been looking at so far. And so one of the things that I'm always curious about, do you think the diet actually impacts your brain directly? I'm not talking indirectly through your gut microbiome. Do you actually think that the gut will directly impact, what you eat directly impacts your brain directly, not through your gut microbiome? And just making sure I'm on the right page. Before we get going, I haven't talked about this yet, but I wanted you to think a little bit about this. So when we are looking at the sucrose preference test, whether or not a mouse will eat something that contains sucrose, a sweet substance, do you think that it is any of the answers down here at the bottom? And we'll see how you do. And we'll let that go for a couple of minutes. We'll see how you guys do. We are going to talk about the sucrose preference test a little bit later on, so it's an important tie-in. And you might want to think about our discussion about depression. So when you're depressed, does it affect some of the basic drives like eating? Does it affect things like sleeping? Does it affect your motivation to move? And does this relate to what we were talking about earlier? And we'll see how you guys are doing. Okay, so increased levels of delta phos B. Okay, awesome. All right, so we'll come back and visit that a little bit later on. There are actually two answers that are correct on here. So the one, we can actually alter delta phos B. It will change motivation and the depression-like state, but it's also the same thing as looking at tail suspension tests, or TST. So if you answered either A or B, great, because we're going to look at both of these in a little bit. Okay, now to go back here. And is it HDMI 1 or 2? 3. 3, okay. All right. So in the last lecture, one of the things that we talked about is we want to understand the neural circuitry. We want to understand the molecules that control the neural circuitry. So it's one thing to know that we have circuits that connect different parts of the brain that are involved in motivation, that are involved in addictive-like behaviors, but we also want to know like when we can turn that switch on and off, or how we can fix that circuit if it's not working. One of those molecules that we looked at is indeed delta phos B. It's a transcriptional regulator, as I've mentioned here in the last lecture. And it's one of those molecules that you'll need to know for the final exam, which is why I'm bringing all of that up for you right now. In addition to that, I also wanted to look at a different molecule. And this molecule helps us to bridge what we talked about in the last lecture with what we're going to be looking at in this upcoming lecture. So there was this really interesting molecule that was found back in 2010. And for the most part, the literature's been pretty consistent. This molecule known as MKP1, this phosphatase that I'm going to be introducing to you in a moment, that was reported in Nature Medicine by Durek et al., was a really interesting molecule. So if you look at depressed human brains, so here again, we always need to go back to the human condition. And in depressed human brains, when we do microarray analysis, can we see that different genes are upregulated, downregulated? And one of the things that was really interesting was that when they examined different areas of the hippocampus, so remember, or you may not remember, but one of the things that is really bad about depression, the mood state is really bad. But for students, what makes it worse is the part of the cognitive brain that allows you to concentrate, to focus, to do things requiring memory tasks, and that's very, very seriously impaired. And so the hippocampus is one of those areas that we've talked about before that is also important to understand in the context of depression. And when we look at two areas of the brain in the hippocampus that are really important and will be a really important part of our discussion today, these are both structures that are key players within the hippocampus. And we're going to see the dentate gyrus a lot today, as well as the CA1. CA1 is sort of where we look at long-term potentiation, learning, memory, and all of those different things. But both of these structures together would be considered to be part of the hippocampus. And when we look at MKP1 and the mRNA, and we look to see, is there an mRNA that is changing and is being upregulated in depressed individuals? If we look age age-matched controls in these black circles, we can see that MKP1 is indeed increased significantly both in the dentate gyrus, not as statistically significant in this particular graph here in the CA1, but it is still trending toward an elevation. And in fact, when you go in and specifically look, not through microarray analysis, but now you do RT-PCR, then you can actually see that MKP1 is significantly upregulated in these areas of the brain. We see it both in the dentate gyrus as well as in CA1. So MKP1 in depressed individuals compared to their age-matched controls is indeed elevated. Are we all good with that? So here's a molecule that's not going down. Here's something that's going up. Are we good? We're good, kind of good, semi-good. You're distracted by chipmunks in my hall, maybe? Okay. I'm assuming that you're all good to go. So again, a really important slide for you to look at in context, and I'm going to ask you a few questions as we go along here as well. So here we're looking at the dorsal hippocampus. We've just identified a molecule, MKP1, that we think is upregulated and is related to depression. So if MKP1 is upregulated in depression, what would happen to a mouse where we inject and then overexpress MKP1? What would your prediction be? We would likely see that the mouse looks like it's depressed. And so here we are using a virus and we are injecting in a virus that also contains GFP. So we want to see that it's being expressed, and we are injecting it into the hippocampus. And within the hippocampus, when we look specifically at CA1, and we look at the dentate gyrus, we can see expression of the adeno-associated virus, which is what AAV stands for, that has been used to deliver MKP1, the gene, that will overexpress this gene in this area of the brain. Okay, so both in the CA1 and in the dentate gyrus. And this green is just different subsections of this hippocampus in these smaller panels to the right. Are we good here so far? So let me ask you this. Why are they bothering to show these pictures over here on the left? On the right. So why are they showing these pictures? So they're showing, obviously, the structure of the hippocampus. You can see the dentate gyrus, or this C-shaped structure. And then when you take a look, we see that the control injection and then AAV that contains MKP1. Why are they bothering to show that? We've injected it, and now they're just showing a structure of the hippocampus. Any ideas? But don't we see that? It's a good answer. Thank you for that. Don't we see that on the left panel? We do see expression in the left panel, right? We see fluorescence. The gene itself is being expressed. Anyone else? Yes, Jen. Right, so there's no change in structure. So overall, the circuit, we're just adding an on-off switch, but we're basically not altering the circuit at all. The structural elements of this circuit, the CA1 looks the same in control as well as in AAV MKP1 injected animals. And so earlier I asked you on that poll that I just put up there a few moments ago, how do we look at this sucrose preference test? And this sucrose preference test is one of those things that we can use to assay or to assess whether or not an animal has become depressed. So again, think a little bit about what depression looks like. So depression causes a change in eating behavior. In some individuals, it causes more eating, and in some individuals, it causes less eating. But overall, if we were to take a look at it, the motivation to eat is actually significantly decreased in most individuals. So sucrose is something animals like to drink more of or take up more of. And one of the things that we're seeing here is this percentage sucrose that is consumed is the normal level is over here. This is what we would expect to see after we do the control injection. So this is what we would normally expect to see. We have not induced depression. We have done nothing to this animal except inject in some of this MKP1 into the hippocampus. And one of the things that we see is that after this injection, and only the injection, we're not doing chronic unpredictable stress, we're not doing foot shock, we're not doing anything else. What do you notice about the amount of sucrose water that this animal is taking up? So it's lower, it's significantly lower. So this again is a sign that MKP1 expression in this area of the brain can indeed cause things that look a lot like depression. So if we also look at the number of escape failures or overall ability to move, one of the things that we would see that normal animals, if they're put into a maze and then they're allowed to escape that maze, will be able to escape without having any difficulty finding the route to be able to escape. So they have very, very few failures, in other words. And again, remember, we're looking at the hippocampus. The hippocampus is involved in learning and memory and spatial navigation. And these animals that now have MKP1 overexpressed, actually they fail to escape many, many more times. So we are affecting their mood and their motivation. That's the sucrose preference. We're also looking at cognitively, they're not doing as well in panel E. And then the last one that we are looking at in terms of their latency to feed, their feeding behavior is also reduced as well. All of these things suggest that the simple overexpression of MKP1, at least in this particular paper and subsequently in other papers, looks a lot like depression. So there's cognitive effects in panel E, there is an eating disorder in D and F, and all we've done is overexpress this virus that contains MKP1, the gene, in a select group of neurons. Are we good so far? Are there any other tests that you would want to do to see if this animal is depressed? Tail suspension test, that would be a great one. Any other ones? Forced swim test, another test as well. So fantastic. So now you know some other tests that could be used, and especially on short answers. If you're looking at depression models, these are ones that you would want to go back in as well as include. Which begs the question, what is MKP1? And why is MKP1 upregulated in depressed individuals? And why does MKP1, when we overexpress it, not following like models of depression, but just overexpressing it, why does it produce all of these problems in these neurons? So if we take a look, and if this was a typical cell, if this was a typical cell membrane, so this is the plasma membrane, and on the plasma membrane, we would have a number of different types of receptors, growth factor receptors. The most important ones for us would be the ones that are called track A, track B, and track C, so track A through C. In particular, the ones that I'm going to be focusing on over the next few weeks are track A, B, and C. And the importance of this is actually very, very profound. So there's a lot that I need you to understand on this slide. And in addition, as you're going through this slide and you're thinking about how to study for the final exam, you're going to see this same pathway 2 other times in today's lecture. And you need to link all of those different times and the context of those different times through the other slides that I'm going to point out to you a little later on. As with most growth factor receptors, whether we're looking at insulin or insulin-like growth factor receptor or the fibroblast growth factor receptors, these are usually monomers, so there's one subunit, and they don't do very much by themselves. However, in the presence of a growth factor, so if a growth factor is around, like BDNF, and we'll talk about BDNF as a growth factor, if a growth factor is present, or if insulin is present for like the IGF receptors, or fibroblast growth factor is present, you need 2 of these monomers coming together, and they form this dimer. And when this dimer forms, one of the things that happens in the intracellular domain of this dimer is that it will autophosphorylate these key tyrosine residues. Okay? And these tyrosine residues are the beginning of how a growth factor will cause different things to happen within the cell. So is that part okay for everyone? So BDNF will work through tract B receptors in particular, but this is just a general talk about growth factor receptors. Any questions on this at all before I go on? Nothing? Nothing, okay. So one of the things that happens is after this autophosphorylation on these tyrosine residues, and if you're wondering what are those Ys, they represent the single amino acid code for tyrosine, as you remember from biochem. This will then activate this RAS pathway. This RAS pathway now gets turned on. This will signal through the RAF, the MEK1/2 pathway, and then it will cause the phosphorylation of a cytoplasmic protein that is crucial called ERK1/2, and they normally will be phosphorylated. This step is important because if this occurs, so growth factor binds, turns on this cascade, activates ERK1/2, it gets phosphorylated on this threonine residue and on this tyrosine residue, it will in turn cause the phosphorylation and the translocation of this molecule known as CBP. And this pathway that I'm showing you is going to be, again, repeated several times in today's lecture. So it's a good pathway to make sure that you're spending time reviewing from either Bio 130 or Bio 230. And it's important in neurons. This pathway here is incredibly important within neurons. This CBP, Krebs binding protein, also works as a dimer. So that's why we have 2 of them. And this Krebs binding protein, or CBP, if it is phosphorylated, its activity increases tremendously. And I'm going to show you on a slide, I think slide 14 or a little bit later on. It gets specifically phosphorylated on serine 133. Okay, and I'll point that out to you again later. Why all of this is important is that if there's been a growth factor that has been activating this cell, this will then allow for the transcription of BDNF, or it will allow for tract B expression from this cell in response to the growth factor, or the production of VEGF, vascular endothelial growth factor, or others to be upregulated. And these are important for how the cell proliferates, whether or not there's going to be more plasticity, whether or not this cell will survive or not. So this pathway is important. And why are we talking about it in this context? Because MKP1 in neurons, so if you overexpress it in neurons, which we just did in the previous slides, or if we activate it through chronic stress, and again, there are multiple ways to do this, but chronic stress is in and of itself enough to activate MKP1 inside the cell, and this is a phosphatase. I mentioned that to you earlier, this phosphatase. And why is this phosphatase important? Because its specific substrate is ERK1/2. And that means that even if there is a growth factor present, if we have overexpression of MKP1, it is enough to block all of these downstream pathways, like cell proliferation. It will block growth factor maturation or growth and maturation, plasticity, etc. And this is why when we overexpress MKP1 in a normal cell, it just starts being aberrant. It doesn't respond normally. Okay, so that was an awful lot that I just threw out at you. Do you have any questions on this or anything that I can clarify for you now? Yes, Amy? So both of these, whenever you see these things in tandem, so both the track A and B, track B receptors will form a dimer, as well as the CBP or the Krebs binding protein, they will also require being dimerized. Any other questions or things I can clarify? Yes, Isis? Yeah, so normally this Krebs binding protein is sort of sitting in the periphery, and it will move from this interface between the nuclear membrane, and it will move down into the DNA and will bind to specific promoter regions. Yes?
So when ORF1 is phosphorylated, it immediately phosphorylates CBP, or does it go through the MSK pathway?
So it does need to go through this risk pathway, and it does need to go through all of that to cause the phosphorylation. So, but you need that in order for risk to be activated, etc. So you do need these pathways here. Anything else I can clarify for you now? Yes.
You said that we need to pass the MSK and the RSK pathway. Do you need both, or is it one or the other?
So it turns out that you don't need both for this to occur, but you do need at least one.
And is one of them like more effective than the other?
It depends on the neuronal cell type. It depends on what growth factor is binding. It depends on a number of different things. Any other questions that I can answer for you? Anything?
Just one.
Yes.
So the overall effect of having increased MKP1 is it an increase or decrease in something else, or is it a neural response?
So if we overexpress or we produce more of MKP1, its effect is to basically block the normal function of the cell. That cell is not going to thrive. It's not going to be functioning normally. All of these downstream pathways will be affected. There won't be production of BDNF, tract B, etc. All of these will be turned off downstream. Expect to see this on the final exam? Yes, very, very excited. Okay, awesome. You probably will, both in short answer form as well as on multiple choice. So before we go on, knowing that this is an important slide, any questions? Yep.
There's an acronym on screen, CUS.
That's the chronic unpredictable stress from last time. So what they're suggesting here is that when they did that chronic unpredictable stress, that in fact they actually did see an increase in MKP1 in those rats within this region of the brain. Anything else? Do you guys know what anhedonia is? Someone can you give me a really quick definition? Lack of pleasurable responses, so perfect. So again, this is showing that you are losing that pleasurable response, and normally animals like drinking water that contains more sucrose. Okay, and by the way, if you knock out MKP1, if you completely eliminate MKP1, those mice where you knock out MKP1 become resistant to developing depression. So again, it's another molecule in your arsenal that you need to be aware of. Okay, so we talked a little bit about the fact that we need to rethink some of our theories, and we are trying to rethink some of our theories. So we're going to talk a little bit more about what all of this means. Some of this is not really clear, so it is really a unicorn. We think it might exist, it might not exist, it might be fictitious. It may not be a real animal, but we're going to try to examine whether or not there are things that are going on. So I mentioned to you earlier, you're going to see this several times. This is that same pathway in a slightly different way. Okay, so the same pathway, slightly different way, and I'm going to emphasize a couple of things to you in this pathway. So one of the things that we looked at is the neurochemical theory of depression is kind of not really clear. There are lots of elements that make it look really attractive, that in fact animal models, if you induce stress, they're going to see reductions in dopamine transmission, serotonin transmission. And we know that if you give different types of antidepressants that work as serotonin reuptake inhibitors, that you have more serotonin at the synapse and they help people with depression. And so there's some things that indicate depression involves neurotransmitters, but there's also a lot of evidence that says that depression cannot only be neurotransmitters. And so that's why we call it sort of a unicorn. Regardless, let's assume for the moment that if we have an antidepressant treatment, whatever it is, like an SSRI, a selective serotonin reuptake inhibitor, that we will actually see an increase at this synapse of things like serotonin or 5-HT, as well as norepinephrine. And one of the things that it can do is that it can act on various membrane proteins, like the beta-adrenergic receptor, and it can actually increase the levels of cyclic AMP, which will then increase the levels of pKa, and then this in turn, downstream, will cause the Krebs binding protein, or CBP, that we saw earlier in the last slide, back in slide number 7. It will cause risk and other factors to phosphorylate them, and we will see dimerization, and we will see them binding to specific regions that will allow for different genes like BDNF to be turned on and upregulated. So this is just a different way to show you the exact same slide. So we think antidepressants actually might work in the same pathway, but not directly, and it might eventually work to increase the Krebs binding protein activation. And again, where we see this phosphorylation is specifically at on the Krebs binding protein at serine 133. Why is that important? If, for example, I told you that I isolated from a cell phosphorylated Krebs, CBP, Krebs binding protein, if I found phosphorylated Krebs binding protein, you know that that cell has been stimulated. It's been stimulated by a neurotransmitter, or it's been stimulated by an antidepressant, or there's been some kind of growth factor added to it. It tells us that the cell is turning on and it is undergoing some kind of genomic change. Okay, so that's key. You need to take those notes down. And for those people who are listening at home, you need to know like the importance of phospho-Krebs. It tells us the cell, especially at serine 133, if it's phosphorylated, it's turned on. And we need to know this for later on in the course. There are also other ways that this could be turned on directly through that MAP kinase pathway. Not all of the steps are shown here, but this is that same MAP kinase pathway that I showed you back in slide number 7. So this makes it confusing, right? So do antidepressants work or don't they work? Do they really work through specific activation of neurotransmitters, and is neurotransmitters enough by themselves, and is that what makes you feel better? Maybe, maybe not, but it's really unclear right now. So before we go on, do you have any questions about this slide? Yes?
On the screen it says phospho-Krebs, as in the Krebs protein itself is being phosphorylated, but you're saying CBP.
It's CBP.
Okay,
Yep.
so it's not the Krebs protein.
No.
That binds
Yep.
to the Krebs.
Right. Any other questions? Okay, so I'm going to take a little bit of a tangent, but hopefully it'll be clear like why we're going on this tangent. And one of the more interesting discussions, especially if you know individuals that have had depression, one of the more interesting discussions that you have is people always want to help, I guess, right? So what would you tell someone who might have depression? Like maybe change the way that you're eating, or maybe go out and get some more exercise. Have you heard that before? Like, oh, go out and exercise. It will cure you of your depression. Heard that before? How about changing your diet? Your diet might help. Yeah, a little bit. Or maybe diet induces depression. So maybe change the way that you're eating. Maybe you'll feel a lot better. And so there's this really interesting sort of link between maybe link between diet as well as depression, and it's really, really unclear. And so does healthy dietary indices, does having healthy food, so I saw that most of you chose fairly healthy food, the apples went, the bananas went, the oranges went, but also some of you are choosing unhealthy food, the Oreo cookies went, the Chips Ahoy's went. And so some of that's healthy, some of it's not healthy, and is healthy food really important? And using that same type of meta-analysis based on observational studies, there seems to be a trend, at least in this meta-analysis and in this review, that says that if you are eating foods that will cause inflammation, so these inflammatory indices, inflammatory diets, DIIs, if you're eating foods that induce inflammation, you're actually more prone to developing depression. Do you like that theory? No. Okay, that was a pretty emphatic no. Does anyone like the theory? I do. Yeah, Lauren? Does it also go with the theory about Parkinson's disease? It could. It very well could. And so do these inflammatory diets lead and link to depression, depressive symptoms? Can it do so directly, indirectly through things like the diet and maybe through the gut microbiome? Again, completely unknown. Like, people are now starting to look at it. And so I wanted us to take a look at that from a neurobiological perspective. How many of you have heard of the journal Nutrients? Some of you have, okay. All right, it was supposed to be one of those questions that no one did. Do you think it's a great journal, high-ranking journal? Would you want to publish in it? Of course you would. You're undergrads. You want to publish in anything. Just get your name on there. Of course you would. It's not a journal that most scientists would want to be publishing in. This is sort of one of the journals that I'm going to take you through. Some of you are asking, like, for your visual summary, do you have to pick like a Nature or Cell paper? Those are great, but you can also choose these types of papers because they give you some insight and different roads to kind of creating future directions of study. So before we get there, I'm going to put up yet another one of these polls. Let's see how you guys do on this poll. So, do you think eating foods that contain omega-3 fatty... So I'm assuming you guys all know what omega-3 fatty acids are? So is it good for your brain? Yeah. It's like this ongoing discussion that I have with my kid about omega-3 fatty acids. So most of you are saying that omega-3 fatty acids are good for your brain. I'm not talking about overall health, just about your brain health. Yes, Izza. This is going through Notonic. That's okay. Don't worry about it. All right, so we'll just go for a couple more minutes. We'll see how we're doing. But do you guys seriously eat omega-3 before every test? You don't really, right? No. You eat fish before every test? So it's like, hey, I have a term test coming up. Let's go eat some sushi. Right. Expensive students. Awesome. All right. Yes? So we would measure inflammation by looking at cytokines, various cytokine markers, and we'll look at that in some more detail. Okay, so I think it's pretty good. So you guys think that omega-3 fatty acids are good for your brain. I would tend to agree. So I had this long discussion with my daughter who doesn't believe that I know anything about neuroscience. She's always kind of like putting me down and saying, yeah, but you know, that's like neuroscience. And what we learn is a little different from what you teach. And so she's a big proponent of like omega-3 fatty acids, and she will go out and buy these expensive omega-3 fatty acid pills, etc. So let's look at whether or not this is actually good for you. So we know that omega-3 fatty acids are indeed pretty good for you in a variety of different ways, especially during development. If you're looking at the treatment of various mental illnesses, many studies have actually suggested that omega-3 fatty acids are good. But then there are lots of other studies that actually say omega-3 fatty acids don't do anything. So again, we're kind of right back to where we have begun. And one of the key things about these omega-3 fatty acids, these are all polyunsaturated fatty acids. I'm going to use the common nomenclature of PUFAs, or for polyunsaturated fatty acids. I'm also going to call these N3 fatty acids, and these are also very much enriched in fish oils, so they're enriched in fish. So having fish, you'll have a lot of these omega-3 fatty acids. These are also known as essential macronutrients. Your body cannot normally produce these by themselves, and so you need to take them in through your diet. So there's a lot of evidence to say that yes, during early development, potentially for mental health, anxiety, mood disorders, these might be a benefit, but there's also a lot of literature, if you look, also saying that omega-3s actually don't do anything. There's no effect or no net effect. And so if we look at this in a little bit more detail, these omega-3 fatty acids or polyunsaturated fatty acids either contain or consist of EPAs or DHA. And they can also have their precursor element, which is known as alpha-linolenic acid, or ALA. And we see these in our diet, and we need to take them in through our diet. These are essential macronutrients, after all, in fatty fish, like salmon. So going out for a sushi before a test, that's great. If you guys want to do that as a sort of control test, you're more than welcome to. I'll pay for your sushi lunch. But I'll also mark your term test harder, so it'll kind of make up for that. You can always try it and let me know if that works, or in those supplements that you can buy as well. The precursor is often found in flax seeds, canola, soybean. Again, you don't need to worry about all of the places that you'll find it in your diet. I do need you to make sure that you know the different types of these polyunsaturated fatty acids like EPA and DHA, and their precursor, which is this alpha-linolenic acid. So again, there's a lot of controversy in this field. So can diet alone make you feel better? So it's not outside the realm of possibility. So think about this. We don't even know how taking antidepressant medication works. And yet when you take it, some people do get better. Not everyone, but about 25-30% of individuals after taking antidepressant medications will eventually feel better after about 3 weeks to about 4 weeks. So there's a lot of logic or anecdotal logic that's associated with taking fatty acids, putting them into your brain. Your brain, most of it is actually composed of lipids. Compared to any other macro molecule, we have more lipids in our brain than anything else. It's incredibly important. I'm not talking myelin, I'm just talking lipids. And so the CNS has the highest concentration of lipids of any of the different organs within the body except the adipose tissue. And this should read organs, not organism. And you don't need to worry about all of the diagram here. I'm going to take you through what I want you to know a little bit later on. But this key idea is that somehow diet might actually influence inflammatory processes. And if we take a good diet, we might be able to induce anti-inflammation. And that's the whole purpose of that nutrient paper that I'm going to go through with you. Can we induce anti-inflammation by taking in these polyunsaturated fatty acids, and can we show that it's protective against things like depression? And again, if we take a look, many of these different types of substances are now thought not to work necessarily on neurotransmitters. So SSRIs, which are these selective serotonin reuptake inhibitors, or SSRIs, or serotonin norepinephrine reuptake inhibitors, SNRIs, these are also thought to work through anti-inflammation. They reduce inflammation in the brain in addition to increasing neurotransmitters. Surprisingly, and I bet you didn't know this unless you're taking like Psych 396 or you're taking neurochemical basis of behavior, that we can also treat depression with a selective serotonin reuptake enhancer. Getting rid of serotonin actually also helps some forms of depression. Does that make sense to you? It sure doesn't, because it doesn't fit our normal theory. But these SSREs that actually remove more serotonin are thought to be involved in some forms of depression. And it also shows anti-inflammation. And of course, omega-3s, these polyunsaturated fatty acids, are also thought to be important in producing anti-inflammatory states, especially within the brain. That's the hypothesis, the working hypothesis. And some of these norepinephrine, dopamine reuptake inhibitors, and SGAs, antipsychotics, these are also thought to work not necessarily on just increasing neurotransmission, but also in producing anti-inflammation. We're rethinking what these are all doing. So it would make sense to at least examine whether or not omega-3s could somehow induce anti-inflammation. If this is what causes depression in the first place, can we reverse that depression by changing our diet? Does that make sense? Is that logical? Okay, thank you. Thanks, awesome. Just love having all this validation from this corner. I feel very validated. Okay, awesome. So just to kind of put things in perspective, we have a lot of lipids. These lipids play an incredibly important role. I'm not going to take you through all of the endogenous endocannabinoid pathways, but the normal endocannabinoid signaling will break down the lipid membrane and allow for some of these signaling molecules that you see here to be expressed, and these can then cross over retrogradely to the presynaptic compartment to affect transmission. You don't need to memorize a single thing on this diagram, but I wanted you to have a feel for how important all of this plasma membrane, which is composed of lipids, how incredibly important that is. We need a lot of lipids within our diet. So again, nothing from this side of the diagram at all. So all of this over here, don't need to worry about it for your own information. But I did want to indicate why we're looking at these omega-3 polyunsaturated fatty acids as this anti-inflammatory agent. Are we good to go? Can I go on to the next slide? Do you have any questions from this slide? Will this slide come back to haunt you on the final exam? Sorry? Only the left side. Okay. Will the left side come back to haunt you on the final exam? Yeah. Yes. Okay. I guess we'll find out on December the 19th. I don't know. I can't say. I don't know. I'm just randomly throwing out dates. I don't know. I have no way to confirm nor deny dates at all. Okay. Hey, I don't make policy. Don't make policy. Okay. So, couple of things here. This is an important figure, so I'm not going to lie. You're going to need to know this figure a lot and in a lot of detail. We've talked about microglia before, and we've talked about markers for microglia, including CD11b. Many of you got really great answers on that short answer question about CD11b. We can also mark microglial cells with another cell surface marker like CD68. In fact, there are a whole host of them. But these are really, really good for marking what we used to call these M1 types of microglia. These microglial cells, in response to their local environment, can either produce inflammation, so they will cause neuroinflammation, or they can cause a healthier environment like neuroprotection. And there are some key molecules that we didn't talk about before that I need you to understand and I need you to know for the final exam. And yes, there will be multiple choice test questions based on this one slide and understanding what's on this one slide. So if in the next 2 minutes when I take you through this slide, there's something you don't understand, please let me know or come and grab me and I'll try to make sure that I do it after the break. So these reactive microglia that produce neuroinflammation, which are pro-inflammatory, which we used to call M1, and we're moving away from this designation, but if you see M1, It's neuroinflammatory, pro-inflammatory, reactive microglia, has lots and lots of the CD11b. We see the short amoeboid cell bodies, etc. And why is this important? These are the ones that are going to be producing that interleukin-6. Remember we talked about interleukin-6 earlier on? Remember TNF-alpha as one of those molecules? This other molecule that we didn't talk about is nitric oxide. And this becomes incredibly important when we start analyzing this nutrient paper. Nitric oxide is released from inflammatory microglial cells. Normal microglial cells, resting microglia, no nitric oxide. They don't produce it. And when we look at protective microglia, they actually have the opposite effect. They actually will take away any nitric oxide. So let's look at the other type, the M2, the anti-inflammatory, the ones that we will produce when we look at the other unicorn, which we call exercise. In exercise, we will be inducing more of these anti-inflammatory M2 type microglia in your brain. And so why is this important? Well, they produce the anti-inflammatory cytokine interleukin-10. They will also produce a molecule known as MRC1 that I'll come back and talk to you about later. It's involved in the anti-inflammatory pathway. And more important than that, it will also produce arginase 1, or ARG1. It's short form for arginase. So it takes arginine and it will cleave arginine, so it's arginase 1. And if we see an increase in arginase 1, we always see a reduction in nitric oxide production. So it's the flip side, exactly the flip side of these reactive microglia. And these types of M2, anti-inflammatory microglia, these are the ones that will also produce more of this magic factor, brain-derived neurotrophic factor, other things like glial-derived neurotrophic factor, and fibroblast growth factor as well. So they're very supportive. They are neuroprotective. We produce more of these after exercise, and they're more beneficial to your brain than the M1 types are. Do you have any questions on that or things that are not clear that I can clarify for you now? I'm guaranteeing that you are going to get a multiple choice test question for sure, and likely a short answer question on this. So this is one of those slides that you circle, you go back over, you think about, you revisit whenever you possibly can. So any questions before we go on? Yes? So if we have M1, will we see an increase in nitric oxide? I'm going to show you parts of the process, but you don't need to know all the details of nitric oxide production. Yes, question? Can reactive microglia be protective? Under very, very limited circumstances following certain types of injury, but generally not typically. So the basal state is the resting state. And then they can either, depending on what's happened, whether it's trauma or whether it's something beneficial, they can convert to one or the other. Any other questions? Yes? Generally, they sit in a transition state, which is the resting state. So they don't convert directly. No. Any other questions? It's arginase 1. Yeah. Any other questions? Yes.
Are they localized anywhere, like for themselves, anywhere particular?
They will localize to an area or specific areas depending on what sort of insult you're doing. But if you're looking at a global change, they will be like everywhere throughout the brain. Okay. Any other questions? No. Okay. Shall we take a break then? Yes, let's take a break. We'll come back and we'll talk more about microglial cells. I don't know. You need to have the access code, which I don't have anymore. So, like, if you ask one of your colleagues, they might have the Notonic activation code.
It shows that it won't even load on the. So it told me to load it onto Microsoft
I would suggest try emailing support@notonic.com, and don't worry about today. Like it's fine. So,
Hey, Professor Xu.
hello.
How's it going?
Good.
So, I was just a little bit confused about phosphocreb. Is that the protein that binds to Creb, or is that the Creb itself?
It's not the Krebs itself. It's the Krebs binding protein. It should be serine 133, CBP 133,
CBP133.
phospho serine 133. Yes.
Oh, so then when you say phosphocreb, you mean the protein that binds to Creb? CBP.
Sure. Yes.
Okay.
Yep.
All right. Thank
Right.
you so
Okay.
much, Professor.
All right.
But I'm just like unsure if like I should just focus on one rather than both because that wouldn't.
Professor Xu, are you saying that the Creb is phosphorylated here?
No, it should be CBP.
Oh, the CBP is phosphorylated.
Yes.
I see. Okay, thank you.
Just no transplant and just
So we'll do your announcement right after everyone gets back.
Okay.
We're just going to tell them like there will be a survey that's coming out that when they see it, it would be useful for your study, and that you're doing a research opportunity 399, looking at note taking, how note taking preferences are changing, how we can improve the experience for note taking, and it'll all be anonymous. It'll be going through you. Like I won't see the data. We're not going to have too many notonic questions, but just like in general, like note taking, studying, and how that plays into like an academic learning. Cool.
Just wanted to share like how specific I was supposed to get.
No,
so
no,
just really
no.
quick.
Yeah. Hello. Good.
So, you said that for the M1 microbial activation, there's increase in nitric oxide, right? Which
Yes.
is pro-inflammatory.
Yep.
But isn't nitric oxide isn't it a ligand for soluble guanylyl cyclase, which produce like
It does a number of different things, and in fact now, the most recent paper that's come out looking at high salt diets. So high salt diets actually cause cognitive impairment that eventually leads to dementia. No one was able to figure out like why that happens, and it's through nitric oxide production,
okay.
and it tends to be detrimental to neurons.
Because I think if I'm not mistaken, activation of the SGC would result in anti-inflammatory effects and like inhibition of platelet aggregations and because of that acceleration.
Okay.
Now we see
Just
the
the
rest.
brain.
Wakiza, you can make your announcement when everyone's back.
Sorry?
You can make your announcement when everyone's back after Chelsea's done hers.
Perfect. Thank you so much. I appreciate it.
Thank you.
Okay, so thanks for your question.
We just wanted to make sure because we were a little confused when we were talking about which one is phosphorylated between the CBP and Krebs. So, which one?
So I'm gonna come back and readdress that.
Oh, okay.
For the time being, the Krebs molecule is important, and the Krebs binding protein is important, but I'll come back and address that in more detail.
Okay. All right. Thank you.
Thanks for the question.
had a really quick question about the visual summary. Just in terms of citing, if we're getting all of our images from BioRender, do we need to cite? Oh. If we're getting all our images from BioRender, do we need to cite each individual one, or can we put like a citation at the bottom? Okay. Thank you.
And I would really, really, really appreciate it if you guys took the time to fill it out and just like major information like regarding your note-taking preferences, how you like to take note, how you think that impacts studying, and like your academic preferences. Can you guys hear me? Sorry? Yeah? Okay. And your academic like preferences, your academic scores, and things like that. So if you have any questions, you can talk to me. I'm right there. But I really, really, really hope you guys take the time out to fill out the poll because it gives me like really valuable data for my research. And yeah, that's it. Thank you.
Hi everyone. So I just wanted to, as the third year rep for NOS, I wanted to remind you all about the event that's happening this Saturday. It is November 2nd, lunch and breakfast provided. You can buy tickets online on our Facebook page, NOS, so Neuroscience Association for Undergrad Students. It's going to be from 10 a.m. to 4 p.m. The topic is neurobiology, so if you really love what we're studying here, which I love too, I really recommend you coming. You're going to learn firsthand about research being done right now by the researchers themselves. So this is like the research that you haven't heard about yet. So please, 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 please show up. It's going to be a blast. I really do hope I'll see a lot of you there. Thank you so much.
All right, thank you, Peter. Thank you.
I appreciate it. Thank you.
To get to where we need to go and understand this paper, there are a couple of things that I need to point out to you. So in this paper that I'm going to take you through, their model of inducing inflammation in the brain is going to be a direct model. So you need to understand this. And what they're going to be doing is delivering what is known as a lipopolysaccharide. And a lipopolysaccharide is part of the cell wall of bacteria. And these are gram-negative bacteria. Again, we kind of talked about that earlier, and those of you who are taking MGY 277, good luck on your weekly quiz. but this also gets into the gram-negative bacteria. Their cell walls, they will have this lipopolysaccharide, and when we deliver it to the brain, they will activate the microglial cells and they will produce inflammation. And how we deliver this is through direct intracerebral ventricular delivery. So we drill a hole in the brain, we put in this cannula, and then we slowly add small amounts of this lipopolysaccharide to induce inflammation throughout the entire brain. This delivery will activate the entire hypothalamic, pituitary, adrenal gland stress-related axis, and it will activate the microglia, which is what we want. I just spent time going through the M1, M2 microglial pathway. If you do this neuroinflammatory model where we are just introducing lipopolysaccharide or LPS, it actually induces a state of anxiety and depression-like behavior. So think, what do I know about assessing depression-like behavior in animal models? So lipopolysaccharide, intracerebral delivery, activate microglia, induce anxiety and depression-like behaviors. And in addition to that, as a bonus, we also see disruption or dysfunction of many of the same monoamine neurotransmitters, things like dopamine, things like serotonin, things like noradrenaline, and we see serotonergic neuronal loss within the hippocampus and within the raphe nucleus. If you're wondering what the ABCD is, I forgot to bring the folders. I have these folders all laid out, and each of these folders indicates a level of difficulty of the final exam. And you are choosing the level of difficulty of the final exam between like A, B, C, and D. So we'll leave that up there for a while. Choose carefully. I remember what the folders are. I'm not changing the order. So I will tell you at the end like what you have chosen for your final exam. And I promise I'm not jigging it or anything. Choose very, very carefully. Now everyone's going to go and plan, and like everybody, let's try to make sure that every single choice is exactly the same. Yeah. It's not going to happen. Okay. So what we want to do okay, let's go on with the lecture first. So what we're trying to do is to understand how these polyunsaturated fatty acids, good job, teamwork, yay, is to try to understand how these polyunsaturated fatty acids, which I told you are anti-inflammatory, how can we test this? So here we have a model that I just showed you on the previous slide, LPS, bacterial cell wall, induces inflammation. Inflammation itself induces. Nice. Right. Okay. So one of the things that happens is that we have this model for depression. It produces inflammation, and our key hypothesis is that these polyunsaturated fatty acids, or the N3 polyunsaturated fatty acids, could actually protect against depression by inducing an anti-inflammatory state. And so one of the things that could happen is that normally these N6 fatty acids can get converted into these other molecules, which are a precursor to pro-inflammatory mediators, and we could potentially block this pathway. We could use these polyunsaturated fatty acids, we could block this pathway from forming, and that might be an anti-inflammatory pathway that we could examine. Maybe that's the way these PUFAs will work. The problem is, in the literature, as I mentioned to you earlier, if you look in the literature, it's about a 50-50 split, much like the choices that you were showing here, although I think A is in the lead, no? Oh, A is in the lead. Right. Hey, maybe it's a bonus, I don't know. So if we can somehow look at this model, if we can somehow look at this model in a clean way where we don't have any of these confounds, because there's a lot of confounds related to, oh, you know what? the polyunsaturated fatty acids that you delivered orally in the food actually didn't make it up to the brain, or most of it was absorbed or excreted, and so we weren't looking at the effects. Or simply other people, when they do it, they can't see the same effects. It's not repeatable, or it can't be replicated. So they want to avoid all of those controversies. Is there a way to avoid the oral delivery of these polyunsaturated fatty acids and to just have these N3 omega-3 polyunsaturated fatty acids in the brain pre-existing without having to change anything else? So the way that these authors tried to approach this is to use a transgenic mouse model. And they're using an enzyme known as FAT1, which I'll show you on the next slide. And this FAT1 enzyme, endogenously, when it is expressed, will allow for the conversion of N6 to N3 PUFAs, the omega-3 fatty acids, the ones that we think are anti-inflammatory, directly in the brain. None of this feeding the animals, none of this making sure that each animal gets the right amount. This is any N6 that exists within the brain automatically gets converted into N3 because now they have this FAT1 enzyme. Okay, so this is key to understanding the rest of this paper. So this is a transgene that's undergone some modifications. Normally, mice do not have this transgene, and this transgene that is being delivered normally comes from the roundworm or the nematode, which is known as C. elegans. And many of you will have heard of C. elegans. And this FAT1 enzyme is an omega-3 fatty acid desaturase. So it converts N6 and it converts it automatically to N3. So it's involved in converting N6 polyunsaturated fatty acids directly into this N3 form, as long as the FAT1 gene is there. And in C. elegans, they have it, so they can convert anything that contains N6 directly into N3 through this desaturase. So the authors in this paper created these transgenic animals, and these transgenic animals will now take that FAT1, normally found in C. elegans, and they will express these now in mice. They normally don't have FAT1. That's why it's a transgene. It's being delivered to these mice. And these FAT1 mice will also be able to convert N6 polyunsaturated fatty acids into the anti-inflammatory N3 type. The wild-type mice, they can't do this. Everything stays as the N6. Is that okay for everyone? We are delivering this FAT1 desaturase, and it will convert any of the N6 directly into the anti-inflammatory N3. And our hypothesis is N3 is protective against inflammation. So these animals, the ones that have the FAT1 transgene, will endogenously, by themselves, convert N6 polyunsaturated fatty acids directly to N3 polyunsaturated fatty acids, and they will maintain these high levels of the proposed anti-inflammatory N3 PUFAs, even if they're just given a diet of like N6. So they convert everything to N3. Clear so far? So N3 is anti-inflammatory. That's what we want. It doesn't matter what diet we're giving them. They will automatically convert it all into this N3 form, which is what we see here. This is sort of the outline of how they did their experiments. So they do the surgery, and they will introduce a cannula, where they will also introduce an inflammatory state by producing or putting in lipopolysaccharide about 2 weeks later. So they inject in LPS. This induces depression-like syndromes, anxiety-like states. It also induces inflammation within the brain. And then they will go back and they will test whether or not these animals, after the delivery of LPS that I showed you earlier, has been implicated after intracerebral ventricular delivery through that surgical route. This will produce depression and anxiety-like behaviors. They test it using all the tests that we now know. So they use sucrose preference tests. Will they drink as much sucrose as a non-depressed animal? Tail suspension test. Will they continue struggling or will they stay immobile? And then the forced swim test. Are they going to swim more often? Does that all make sense? Is there anything that I can clarify for you now before I go on, or anything that, yeah? Yep, they will have 4 different groups. So they'll have a group of animals that have control, that are FAT1 overexpressors, etc. And each of these groups will have like 6 different animals in them. Any other questions that I can answer for you now? No? Okay. So here's some of their experimental data. So I'll take you through the most important parts of this. And here, again, they're looking 1 day after, if we look at the experimental timeline. So they do LPS, they inject this in, causes inflammation. They then wait 1 day, do the sucrose preference, tail suspension. 2 days later, 3 days later, they'll do the forced swim test. So here, do we see signs of depression? So do we see a reduction in sucrose preference? So this is the normal level. So these are the wild types. They get injected with saline, no inflammation. These are the FAT1 overexpressors. And the FAT1 overexpressors, they actually see, after the injection of saline, they look identical to the wild type controls. And then if we look at the wild type, where we have injected in LPS, intracerebrally through the ventricles, they will have inflammation, and we also see signs of depression. They don't drink as much sucrose-containing water as the wild types over here do. Surprisingly, the mice that have FAT1 that will then convert N6 to N3, the anti-inflammatory polyunsaturated fatty acid, even if they're given an inflammatory molecule, they don't look like they're depressed. Is that clear for everyone? So at least on this one slide, it looks like it is protective. Having polyunsaturated fatty acids through the conversion of N6 to N3 will be protective against this form of depression. So 1 day later, while they're still in this inflammatory state, they look for immobility. So that tail suspension, how much time are they spending where they're just staying still and not struggling? The longer they're staying still, the more immobile they are, the more likely they are depressed. Okay? So again, the wild type saline is our control, and we see that they are staying still a certain length of time. The FAT1, saline delivery, no inflammation, they will struggle for the same amount of time. The wild type LPS, they stay still much, much longer. So instead of staying still for this length of time, there's an increase in their immobility time that you see here. And this goes down if we inject LPS into the FAT1 anti-inflammatory mouse model. So do both of these tests agree with each other? Yep. Okay, I would agree to agree. Oh, so sorry, your class blockade is slowly dying off here. Okay. All right. So if we continue on, there's a couple of other things that I need you to be aware of as well. In this particular paper, because it's a nutrient paper, it's not sort of the high caliber, high quality paper that you would normally have for like Nature or for Cell or any of the top tier journals. They actually don't do the types of stainings that we would want. They don't do CD11 staining or CD11B staining and then try to compare them back. They don't look at morphology, etc. And one of the things that they look at is these molecules here. Do you remember nitric oxide? Do you remember what type of microglial cells produce nitric oxide? So it's the M1, the pro-inflammatory type. And the thing that actually gets induced, and this is why we have the iNOS at the top, within those microglial cells, the ones that are pro-inflammatory, that are producing more of the nitric oxide, and one of your colleagues, and I forget who it was asking, do we need to know all of the steps in the pathway? I told you no. No, nitric oxide. Hey, that's cool. All right. Nitric oxide is inducible. The synthetase is inducible. iNOS is inducible. And we can see whether or not we have more in the brain tissue or not. And again, here they're just taking the entire brain tissue out. They want to know, in terms of enzymatic activity, do we have more nitric oxide synthetase activity in the brain tissue or not? Wild type saline, we have this basal level. We don't expect to see an increase, but we see the fat saline levels are about the same. And we see an induction of the nitric oxide synthetase only after LPS delivery. So we see more activation, which could correspond to microglial activation. They're not measuring it directly here, but you would have to assume that this is related to microglial activation. And then here, the FAT1, the anti-inflammatory pathway, is activated, and we see a reduction, right? So back to baseline levels. This is where the data is not so clean, but also statistically significant. So if we look at the wild-type saline, and we look at the wild-type LPS, we see that we get an increased production of nitric oxide, and it's very difficult to measure this, by the way. So you're doing a biochemical assay for this releasable gas that you're trying to measure in minute quantities, and it's a very tough biochemical assay, but they do see an increase versus the wild type. So there is the activation of the inflammatory pathway. Lauren, did you have a question? Yep, it is involved in a number of other processes. In this particular case, they're concentrating on nitric oxide specifically to make the case that this is an inflamed brain, and they're reducing inflammation by having the N3. But it is involved in a number of different processes. And for those of you who are interested, there is a paper that came out last week in Nature that actually looks at nitric oxide, the effect of diet. It's been a mystery for a full year, but a group had come out earlier to suggest that if you have a high salt diet, this high salt diet and a continued high salt diet will actually cause cognitive deficits and will eventually cause dementia in mice models. And they weren't able to figure out the mechanism, but they did release a second paper that came out last week that actually implicates nitric oxide in that whole process of neurodegeneration. So it's likely involved in a number of processes, Lauren. Here they're just trying to keep it fairly simple. Okay, so there's a lot. Any other questions before I go on? Or anything that's not clear on these graphs or slides? No? Okay, so a couple of other things that I'm going to mention because this will be a little bit important later. Do you remember one of the earlier diagrams that I showed you that one of the key players, and this is going to be a key player for the remainder of the course when we talk about exercise, cognition, brain power. Whenever you're talking about a healthy brain, we're always interested in like BDNF. So if anyone like comes up to you in an alley and says, hey, do you want to buy some BDNF? You always say yes, right? So you take it. It doesn't matter like how much it costs. You buy that BDNF. And here what you're looking at is that in the case where we have inflammation in the brain that is caused by this lipopolysaccharide delivery, without the protective effects of the N3 PUFAs, we actually get a dramatic and significant reduction in the levels of BDNF. Now, I'm not going to talk about pro-BDNF yet. We will talk about it later. And its pro-BDNF is elevated, and it looks like that makes no sense. In fact, after next week's lecture, this should make perfect sense to you. Like, why is pro-BDNF? Isn't pro-BDNF like the bigger version of BDNF? Why is this up and this down? And why is this like a bad thing and why is this a good thing? It will make sense after next week's lecture, and so will this graph here about P75 receptors. But the more important one that I want you to concentrate on is this panel here. We are seeing a reduction in the receptor for BDNF as well, the TrkB receptor. And it is dramatically reduced when it's compared to the wild type. And if you're asking, well, isn't this also reduced? It is, and they don't really address this, and it's one of those things that's a cause of concern, but it isn't statistically significant. The main point being that LPS causes a reduction in both BDNF, inflammation causes a reduction in BDNF production, and the BDNF receptor itself. And those will both become really, really important when we talk about exercise, your brain, whether or not your brain responds to BDNF, why BDNF is so important, and why pro-BDNF is not something you want to encounter. So any questions or anything that I can clarify for you now before we go on? Yes?
How are we supposed to account for the FAT1 saline reduction?
The fat ones, which panel are we looking at? Panel B, yes, and
So fat one, say like mice have an elevation in
so why do these mice also have like an elevation in nitric oxide? It's a great question. So one, they don't really address it, and two, it's not statistically significant, but the trend is definitely there, right? So I would agree with you that it's a little troubling, but their story and hypothesis is really centered around that third bar, which is the wild-type LPS, and that's what they want you to concentrate on. And that's why it's probably published in Nutrient and not in something like Nature, because this wouldn't survive like the editors in Nature. So, yep, good point. Anything else that I can indicate for you now? No. No? Okay. All right, so I'm going to try to make some sense out of this story, because it's very confusing in a lot of ways. We don't really understand depression. It's one of those disorders that, it's going to affect so many individuals, and we know little bits and pieces of it, but we can't make a cohesive, complete story, and so many people are studying it, and yet there's no agreement on it. And it's one of those very, very frustrating elements in science where all of these things just really don't give us like one common mechanism that we would all really want to have. And so I mentioned to you earlier that there's different ways for us to kind of approach what depression is. And again, this is one other avenue of research that people are looking at. It's not really clear like how all of this works either, but this is yet another sort of form of evidence against these neurotransmitters. And I'm not going to be able to give you the whole story, but I'll give you bits and pieces of it. These are two different mice that you are looking at here on the left. So there's this normal-looking mouse, and then there's this mouse that has this band of white tissue or white hair across its backside. And one of the things that has happened is the melanocytes, the cells that are normally dividing and causing pigmentation or the coloration of its hair, have been killed. They've died off. And the reason why it has this little band is this mouse has been exposed to ionizing radiation at very, very high doses. And this has been able to penetrate through the skull, through the follicles, the hair, and it has killed pretty much anything that it's been in contact with. And that's okay. Melanocytes, they're not going to be able to produce more melanin. They're dead. They're dying. The neurons are post-mitotic. They're not dividing anyway, so those neurons are going to stay nice and healthy. Any dividing cell will die off when exposed to ionizing radiation, but neurons are post-mitotic. They don't really care. But these experiments that were done early actually suggested that there's a group of neurons within the subgranular zone layer, this SGZ, this subgranular zone layer, that are continually dividing even within the adult. Now, this became important, not originally, not specifically for depression, but just in terms of neurobiology, that, hey, we have this population of neurons, not in early development, but in the adult, that continue to divide, that continue to form new neurons, and they're found within this area of the brain, and we can kill them by using ionizing radiation, and we are only exposing this area of the brain. These lead plates protect the rest of the brain from this. And in fact, there's a lot of evidence to suggest that in animals, there are various areas, at least two, perhaps more areas in the brain, where we have continuous generation of these progenitor cells, and then they will go to the subgranular zone layer within the hippocampus. And we also have another that is found in the subventricular zone that I haven't shown you yet, and I will a little bit later on. And one of the interesting things about this whole process is that when scientists decided to look at it step by step, when enough scientists believed that this actually occurred, one of the things that they found was that from the time a new neuron is dividing and is born in this subgranular zone layer within the hippocampus, to the time that they start to differentiate, they start to migrate, and then they integrate within the hippocampus, within the dentate gyrus of the hippocampus, this entire process takes about 3 weeks, 2 to 3 weeks. And they were quite surprised. And then they started to examine this in a little bit more detail. So need to ask you this question. Do you need to memorize this slide? No. Why no? This is like one of those perfect slides. Lots of data, lots of things I could test you on. No? So yes. So no. Okay. You don't need to memorize this slide. This isn't an important one. But I am going to point out that when we look at proliferation, all the way to when it gets incorporated, it's about 2 to 3 weeks. Now that time window is actually kind of unique. And this time window suggests, because we know one area of the brain that's affected, and we looked at that with MKP1, is the hippocampus. We know the hippocampus is affected in depression. And in depression, there's a problem with memory, cognition. And here's this thing suggesting that the hippocampus has this unknown mechanism that wasn't known before that takes about 2 to 3 weeks where there's a birth of new neurons. No one had thought that maybe something is happening to new neurons being produced after depression has occurred. And again, it's very, very systematic, very, very well described, and it takes this 2 to 3 weeks. We can actually match every single step using all of these different markers, which you don't need to memorize, that's on this particular slide, and we can see this circuit developing in real time. So it's amazing, and people started to think, hey, wait, maybe this circuit is important in depression. Maybe that's why it takes, from the time you take an antidepressant to when you start actually feeling better, maybe it's helping this process of neurogenesis. Maybe it's actually helping this neurogenesis process, whether it's speeding it up, stabilizing this process. Maybe that's why it takes this 2 to 3 weeks, that mechanism that we didn't know ahead of time. So just a couple of things related to that. So it's been known since about 1997 that there's a decrease in neurogenesis, this area of the brain in the hippocampus, in animal models with induced depression. So induced depression is something we have to use. We don't have a genetic model. Chronic unpredictable stress, that's an induced model of depression. High levels of corticosterone injection, induced model of depression. And when you do that, you dramatically decrease neurogenesis. So they're kind of linked, at least since 1997. And in fact, if you started delivering antidepressant medication, particularly ones that are selective serotonin reuptake inhibitors like Prozac or fluoxetine, you can actually reverse some of that loss in neurogenesis. You can compensate for that loss in neurogenesis. So stress is now thought to directly impact neurogenesis, directly impact neurons within the hippocampus. And one of the ways that it does this is it's thought to reduce BDNF. And that goes all the way back to what we talked about earlier on. BDNF is affected by stress. We saw that through the MKP1 pathway. And we know stress activates MKP1. It turns off the ERK1/2 pathway. It then stops more of the BDNF from being produced. And that might account for this decrease in BDNF. And that might affect the neurons that we see within the hippocampus. Antidepressant therapy, though, or antidepressant medication, things like Prozac or fluoxetine, over time will actually enhance BDNF. It will also increase the levels of these neurotransmitters. And as I showed you earlier, these neurotransmitters and antidepressants also work through those same pathways. Not directly. It might work through the beta-adrenergic receptors, and it may work through cyclic AMP and protein kinase A, or it might work through MAP kinase through a G-protein coupled receptor. Either way, these antidepressant medications might also do exactly the same thing, enhance BDNF, this molecule that we're slowly working our way to. In fact, some studies have actually started to think about using BDNF as a biomarker. It hasn't been widely adopted because some people find that BDNF levels are low in depressed individuals. Some people see no difference. Some people actually see an enhanced level of BDNF in depression. So it hasn't been universally adopted, but some studies have indeed linked low BDNF with depression. And so we're going to look at whether or not one thing that is known to induce BDNF, like there's no doubt that this is not a unicorn. If you exercise, immediately within like a few hours, your levels of BDNF are going to dramatically increase. So that's why people think exercise is like really great for depression, but we're going to look at that after we finish this particular section. So this BDNF might play a really, really important role. And this BDNF probably does so in a number of different ways. We'll look at exercise when we come back from break, but we also know that many endogenous factors, things that normally exist within the brain, can actually do this directly. So if we enhance the release of neurotransmitters, these monoamines, things like dopamine, norepinephrine, things like serotonin, this can actually increase the activation of BDNF. And I showed you that earlier on as well. And so these increases by neurotransmission that cause an increase in BDNF, that takes time, it's transcription dependent, it's also going to be like signal dependent, and it's going to not work immediately. This might be the mechanism by which these antidepressants, by elevating these levels of neurotransmitters, might work. It may enhance BDNF, it may enhance neurogenesis. So going back to this slide, is this one that you should know? Sorry, yes. So just to be clear, this one is a firm yes. This one is a firm no. But I do expect you to know different parts of this particular pathway. We have looked at this already twice today, and so you know that this is an important pathway. And one of the things that I mentioned is that this molecule, BDNF, incredibly important for neurons and their normal survival and differentiation. They will work through receptors that are found on the cell surface, these TRAC receptors. And for BDNF, as the name suggests, TRAC-B, B for BDNF, when BDNF binds, it will activate these TRAC-B receptors, cause their autophosphorylation as they dimerize, and these tyrosine residues will then activate other signaling molecules within the cell. And these different pathways, as a result of this dimerization, tyrosine phosphorylation, will now be activated where it wasn't activated before. So we've already seen this pathway down here, this MAP kinase pathway, this ERK pathway, the activation of ERK, and then the transcriptional activation of things like BDNF, etc. And we haven't looked at this pathway. And this pathway is kind of a neat one for neurons. And if you're thinking about this pathway actually suggests something about neurons. And then I'll leave that open for you, and you can tell me what you think about this. So if we look at this pathway here, we've activated through BDNF or through NGF, something has activated this SOS GRAB2 pathway. The SOS GRAB2 adapter will then allow for the activation of PI3 kinase. And this PI3 kinase, in turn, will activate AKT, which is another kinase. And this AKT, after this growth factor has bound, will actually inhibit all of these proteins that you normally see here in red. And some of these proteins are ones like BAX, things like BAD, and these are all pro-apoptotic proteins. So what does this mean for you? Like, think a little bit about this. These are being turned off by AKT. And these are in neurons. And we're turning all of these cell death pathways off. What does that mean? Any ideas? Yes. It affects cell survival. One more step. So neuroprotective. One more step, even worse. Not cancer. That's too many steps. Take a step backward, back a few. Anything else? So your neurons, by default, and this is true, your neurons by default are always ready to die. They're always just on the verge of committing apoptosis. So, but honestly, think about that. If you don't have a continual, steady supply of these growth factors, and you're not activating AKT in your neurons are going to die. And so this is why these growth factors, and if you can, by the way, one of the speakers that's going to be at the everything N is David Kaplan. David Kaplan is one of those neurobiologists who helped to elucidate this apoptotic pathway. You can ask him more about this personally, because he's found many of these different factors. Your neurons are always ready to die. Without these growth factors there in small quantities, your neurons are not going to be surviving. And so this is the importance of having BDNF, why we're talking about neurotransmitters. So the more active your neurons are, the better your neurons are going to be, because there's more BDNF around, and you're also going to be activating more of this AKT. The last pathway is this phospholipase C gamma pathway, where we're going to produce more of this intracellular calcium, and that's the major pathway that I want you to know. You don't need to worry about all of these intervening steps. In fact, you don't have to know about all of these different molecules in these pathways. The major one is the activation of AKT through PI3 kinase after the TRAC receptors have dimerized, and that's the most important part of it. Okay, so anything that I need to clarify for you about what you need to know or what you're responsible for on this slide before I go on? No? All right, so I'm just going to go through a couple more and then we'll sort of wrap it up. I know I haven't finished all the slides, but we'll come back and do that after. Important slide or not an important slide? So it is an important slide. Most students kind of skip through it. I'm going to tell you that next week is where I'm going to be putting together your final exam. So I'm going to be sitting down, trying to figure out how many lectures I'm going to get through, what I'm going to cover. I'm going to start writing the final exam. And what you're telling me is, so the choices, by the way, do you want to know what the choices were? Okay. Choice A was hard. Choice B was WTF, what the hell was that? Choice C was tear streaming, and then choice D was fair. So since there doesn't seem to be much of a consensus, I'll take a little bit of each. Is that okay? No, of course not. No. All right. Right, okay, good, all right, good. I think fair is the way that we always want to go. Right. Okay. Good. Nice, nicely done, nicely done. All right, so a couple of things on this slide that you do need to know for your final exam. So there are a number of these different growth factors. So these growth factors like the vascular endothelial growth factor, which is incredibly important within the brain. BDNF, brain-derived neurotrophic factor. Again, I've already mentioned how important that is. The insulin-like growth factor 1. In neurons, this is a really important growth factor. And then fibroblast growth factor, or basic fibroblast growth factor, which is FGF2. FGF1 is acidic fibroblast growth factor. We know that neurotransmitters, antidepressants, all work to increase these levels. And we know that stress actually works to decrease the formation of VEGF. It will decrease the formation of BDNF. And why that's important is that neurogenesis within the brain is actually dependent on both VEGF and BDNF. So when you have stress induced by depression, you are going to stop proliferation and you're going to stop neuronal survival. And so this is why we have a reduction in neurogenesis after we have an induction model of depression. And again, very, very well sort of maintained. And why this slide is particularly tricky for students on the final exam, there's a lot sort of on this slide. So if you look, and you weren't really paying attention, do all of the receptors that you see on this slide, do they dimerize? Do you have like two subunits? So all of them. IGF-1 forms a dimer, so does FGF2, BDNF. They all have tyrosine residues, so those little Ys that you see on the inside. So if you see tyrosine phosphorylation, that means the receptor has been activated. And again, keeping that clear. And as a result, we can see all of these different types of changes. The last thing to indicate for you, VEGF and BDNF, they have very different sort of functions. VEGF is more important for proliferation of cells. It causes them to divide much more rapidly, particularly in the subgranular zone. And BDNF although very important for neurogenesis, after they've proliferated, if you don't have BDNF, they die off faster. So even if you've been exercising, if you have no BDNF, you might produce a lot of neurons, but none of them will be left over without this BDNF around. And we'll come back and talk about that later. So just a couple of quick reminders before we finish for the day. So this is the last slide I'm going to cover. Next week we don't have lectures, so I'm hoping that you'll all take a little bit of a break, not too long, a couple of days. I am going to be around most of next week. The only day that I'm not available is Thursday. I will have more test viewing times for those of you who want to come in. Please start thinking about how you're planning your month of November. November is not going to be a favorable month for most of you, and you need to start planning ahead in advance for that. In addition to that, just remember that quizzes will start tomorrow night, 8 p.m. They'll be available. They'll be available for the entire month, but it's better if you do them early rather than leaving them. I will also be posting some extra questions or an extra copy of the final exam for you to start practicing as well. Have a wonderful November break or fall break, and I'll see you all back in 2 weeks' time. All right, so take some chocolate on your way out, and for those of you who are staying for pictures, we'll meet up here at the front.
Do you have office hours during week?
Yeah, I just mentioned, I do. I'll be posting those. You're gonna send me that as well? Okay, awesome, thank you. Yeah, please grab some.